Okay, for any of us in the pharmacy world, getting a handle on pharmacokinetics and biopharmaceutics is, well, it's everything. It's not just about passing exams, it's the science that connects a pill to a patient's outcome. So in this explainer, we're going to do a deep dive into those core principles. Let's get right into it.
So let's start with the big question, the one that really drives everything we do. How does one little pill actually turn into a result we can predict and count on? The answer is all about the incredible journey that drug takes inside the body. And that's exactly the journey we're going to map out today.
Here's how we're going to break it down. We'll kick things off with biopharmaceutics. That's where the drug's formulation really sets the stage. Then we'll follow its trail through the body with pharmacokinetics, the whole ADME framework. After that, we'll get into how we use models to create smart dosing strategies. And finally, we'll tackle the real-world messy stuff, drug interactions and genetics, where our clinical know-how is absolutely crucial.
All right, first up, biopharmaceutics. You can think of this as the first major hurdle. It's all about how the drug's physical and chemical makeup and the way it's formulated decides whether it can even get into the body to begin with. For a regular oral tablet, it's not like the drug just instantly appears in the bloodstream. It's a three-step process. First, the tablet has to disintegrate into smaller bits. Then, the drug has to dissolve from those bits into a solution, and only then can it be absorbed through the gut wall. Now, for a lot of drugs that don't dissolve well, that second step, dissolution, is the real bottleneck. It's the rate-limiting step that controls how fast the drug can even get to work. And the drug's physical form on a molecular level has a huge say in that dissolution step. A drug can be in a messy, non-crystalline state, we call that amorphous, or it can be in a super-organized crystalline structure, a polymorph. So here's the key takeaway. It's way easier for a drug molecule to break free from that messy amorphous structure than from a rigid crystal. And that means the amorphous form is always going to be more soluble, which can be a total game changer for getting a drug absorbed properly.
And this table here, this is where it all comes together. The Biopharmaceutics Classification System, the BCS, is this brilliant framework that sorts drugs based on two simple things: how well they dissolve in water, solubility, and how well they can cross the gut wall, permeability. Just by knowing a drug's class, we can predict what's going to happen. Take a class 2 drug, for example. It's got low solubility, but high permeability. Right away, we know the problem is going to be dissolution, and that's what we need to tackle in the formulation.
So now the drug is successfully unlocked from its dosage form. This is where biopharmaceutics passes the baton over to pharmacokinetics. Now we can start to actually map and predict how much drug is in the body at any given time. So what is pharmacokinetics officially? Well, it's the science of the rates of drug absorption, distribution, metabolism, and excretion. You know it, you love it, ADME. At its core, it's the story of what the body does to a drug, from the second it gets in until the very last molecule is gone. And this graph right here is our main map for that journey, the plasma concentration time curve. Every part tells a piece of the story. Cmax, that's the peak concentration, tells us about the intensity. Tmax is the time it took to get to that peak, which gives us the rate of absorption. And the total area under the curve, or AUC, that represents the total exposure to the drug. Our job clinically is to keep that concentration in the sweet spot, the therapeutic window. That means above the minimum effective concentration, the MEC, but well below the minimum toxic concentration, or MTC. Let's just quickly run through ADME, starting with A for absorption. This is what we were just talking about, the drug getting from where it was given into the bloodstream. And like we just saw on our map, the speed of absorption is all about Tmax, and the extent, how much actually made it in, is measured by the AUC. Next up, D for distribution. Once the drug is in the blood, it doesn't just hang out there. It travels out to the body's tissues. But here's a critical point. A lot of drugs love to bind to proteins in the plasma, like albumin. And that drug-protein complex, it's too big to go anywhere. That means only the free, unbound drug is actually available to go into tissues, find a receptor, and do its job. The bound drug is just riding around as a reservoir. Then we've got M for metabolism. This is just the body's way of chemically changing the drug, usually to make it easier to get rid of. The liver is the main chemical plant for this. And for drugs you take orally, this leads to something called the first pass effect. After a drug is absorbed from the gut, all that blood goes straight to the liver. So the liver gets the first pass at metabolizing the drug, before it even gets a chance to circulate through the rest of the body. This can wipe out a huge chunk of the dose right from the start.
And finally, E for excretion. This is it, the final, irreversible removal of the drug and its byproducts from the body. The two main exit routes are through the kidneys into the urine, which is renal excretion, and through the liver into the bile and out in the feces, which is biliary excretion.
Okay, so we've mapped the entire journey, but how do we actually control it? This next part is all about taking those ADME principles and using them to design dosing regimens that are going to be safe and do what they're supposed to do.
To make sense of it all, we use models to simplify the human body. The simplest one is the one-compartment model. We basically pretend the body is just one big tank of water. The drug goes in, spreads out instantly, and starts to get eliminated. This gives you a nice, straight line on a semi-log plot. But for drugs that take a little longer to get into the tissues, we need a two-compartment model. We separate the blood from the tissues. And that explains perfectly why you see that two-phase drop, a fast dip as the drug distributes out, followed by a slower, steady elimination phase.
Now, let's talk about what is probably the single most important clinical PK parameter, clearance. It is so important to remember that clearance is not an amount of drug. It's a volume of plasma that gets completely scrubbed clean of the drug every minute or every hour. Think of it as how efficient the body is at removing the drug. And its number one job for us clinically? It's what we use to calculate the maintenance dose a patient needs to stay at the right level.
And that leads us to half-life, the time it takes for the drug concentration to drop by half. The formula shows it's tied directly to the elimination rate constant, K. Clinically, this one value is incredibly useful. It tells us how often to give the drug, that's the dosing interval, and it tells us that it will always take about 4 to 5 half-lives for a drug to build up and reach a steady state in the body.
But of course, patients are not simple models, right? This is where our expertise as pharmacists really comes into play, navigating all the complicated stuff where individual patient factors can completely change the rules of the game. One of the biggest complexities is drug interactions. The cytochrome P450 enzyme system in the liver breaks down most of the drugs we use. And as you can see here, other things can interfere. A substance can inhibit an enzyme. The classic example is grapefruit juice blocking CYP3A4. This is like putting the brakes on metabolism, causing drug levels to climb, which can lead to toxicity. On the flip side, something can be an inducer, like carbamazepine, which tells the liver to make more enzymes. This hits the gas on metabolism, which can chew through another drug so fast that it fails to work. And it's not just other drugs. A patient's own genetics can completely change the metabolic picture. This is pharmacogenetics. For certain enzymes, some patients are poor metabolizers. They basically don't have a working enzyme, so a standard dose could be toxic for them. Others are ultra-rapid metabolizers. They have extra copies of the gene, and they might clear a drug so fast that a standard dose does absolutely nothing. And finally, there's nonlinear pharmacokinetics. This happens when one of the ADME processes, usually metabolism, gets maxed out. It just can't work any faster. The textbook example is phenytoin. Once you saturate its metabolic enzymes, even a tiny increase in the dose can cause a massive, out-of-proportion jump in the drug level. This makes things really unpredictable and dangerous, and it's why those drugs need super careful monitoring. So at the end of the day, getting all of this, from how a tablet dissolves to a patient's genetics, is what takes us to the next level. It's the science that backs up every single clinical decision we make. It lets us truly manage a patient's drug therapy to get the best results and keep them safe. And that kind of leaves us with a final thought. As genetic testing gets more common and cheaper, we're really heading into an era of personalized medicine. So does that mean the whole idea of a standard dose for everyone might one day just be a thing of the past? It's definitely something to think about.